I'm Leland Russell. This summer I did work in the Robbins lab using remote sensing for predicting alfalfa growth. And so remote sensing is when we use images taken from maybe rovers or satellites to kind of predict the growth of plants. And so we use vegetative indices, which are a combination of parts of these images. So for example, in figure 1A, we see NDVI, which is one we use using the near-infrared and the red band. And we see in figure 1B that healthy plants often have higher near-infrared and lower red, indicating they would have higher NDVI. It's also generally known to correlate well with biomass accumulation. And so what we did is we had a field which we had harvests on throughout the year, and then we also took images using an unmanned drone. And we created mixed models for genetic correlation and heritability, and then we created hierarchical models, Bayesian hierarchical models, to try to predict that growth. And so generally looking at our data in figure 5A, we see that NDVI and yield have good correspondence, but generally NDVI kind of happens before yield. And so we'll get into that up here. And so we also created heritability estimates, which is kind of a measure of how good and how reliable our data is. And we see that for the most part, we have moderate heritability. We expect it to be higher when we increase our data size, though. And so for our hierarchical models, that's a way of kind of combining assumptions with data to try to form a more robust model. And so we assume that it's logistic growth, which has some parameters which we try to fix or we try to predict using the model. And so when we do that with NDVI and yield, we see a very good fit. But then if you remember down here, there's some time offset. And so what I did is I fixed these three parameters, adding in a time offset and a scaling parameter, and created a model for yield. And we see that they're almost identical, which is very good for us. And so for the future, what we want to do is we used just a subset of this field. And so we want to expand our analysis to include all cultivars in the experiment. We also want to use a genomic relationship matrix, which is kind of a way to measure how similar genotypes are to each other, to try to improve our models. We want to apply this Bayesian framework to different types of crops, say maybe corn, or crops under diverse conditions. For example, maybe drought stress. And then we want to optimize our priors, which is what we're trying to assume, to ensure good fit. And so here we see in genotype 5, there's not the best fit, and so we're trying to work that out. And so we see NDVI shows promises of proxy for biomass accumulation in alfalfa. Larger data sets are generally needed to produce more robust models. But the hierarchical modeling framework shows very stable estimates despite having a small sample size, especially compared to our mixed models.
Well, you did a great job. It's very cool work. Thank you very much.